Hello everyone. Picture this. A young cricketer collapses on the field during a scorching Delhi summer, gasping for his breath. His muscles scream for oxygen, a demand his body struggles to meet. This, my friends, highlights the critical role of hemoglobin and myoglobin, the oxygen delivery dynamos of our bodies. Think of them as the oxygen express, constantly shuttling this precious cargo from the bustling Mumbai of our lungs to the remote villages of our tissues. Why is this so crucial? Why is this so crucial? Well, imagine trying to dissolve sugar in chai. You can stir all you want, but only so much will dissolve. Similarly, oxygen solubility in blood is limited. Without hemoglobin, our blood would be like weak, unsatisfying chai, unable to deliver the oxygen kick our cells crave. Myoglobin, on the other hand, is like that extra sugar cube you keep tucked away for emergencies. What? Only me? Okay. A local oxygen reserve within our muscles which is ready to be deployed when needed. The story of these oxygen chaperones begins in the late 1800s when scientists first observed the unique oxygen binding properties of blood. Think of the early days of Indian railways, slow, chugging along, but laying the foundation for a vital network. Over the years, brilliant minds like Max Perutz and John Kendrew painstakingly unraveled the intricate structures of hemoglobin and myoglobin, earning them the Nobel Prize in 1962. Their work, like the high-speed trains of today, revolutionized our understanding of how oxygen travels through the human body. Now, we are going to start with the key concepts, okay? So, heme, you can see heme as the oxygen magnet. At the heart of both hemoglobin and myoglobin lies heme, which is a ring-shaped molecule resembling a tiny molecular stadium. Nested within the stadium is an iron ion. And if you remember your chemistry days, iron is denoted by Fe2+. The star player responsible for capturing and releasing oxygen is basically iron. This iron, like a skilled cricketer, catching a ball, reversibly binds oxygen. The porphyrin ring surrounding the iron acts as a protective cage, preventing the iron from being oxidized to Fe3+, which can't bind oxygen. So we have Fe2+, which is the normal type, and Fe3+ is a type which can't bind oxygen. This will make sense more in the future. Chemically, the iron forms a coordinate covalent bond with oxygen, which is why it can hold it. This is like a delicate embrace that allows for easy on and off access. Now imagine if that cricketer fumbled the ball. That's what happens in methemoglobinemia, where the iron is oxidized, rendering hemoglobin useless for oxygen transport. This can be caused by certain drugs or genetic defects leading to cyanosis, a blue discoloration of the skin. Now we come to myoglobin. Myoglobin is the oxygen hoarder. Okay, so myoglobin residing within muscle cells acts as a local oxygen reservoir, much like the water tanks we have on our rooftops. It's a monomer, a single protein unit with one heme group. That's it. Its oxygen binding curve is hyperbolic, meaning it quickly saturates with oxygen even at low partial pressures. Now we are going to touch upon the oxygen dissociation curve later on, but just know this for now that the the oxygen binding curve for myoglobin is hyperbolic. Think of it as a thali at an all you can eat buffet. It fills up very quickly, right? This high affinity ensures that myoglobin effectively snatches oxygen from hemoglobin in the capillaries and stores it for times of high demand, such as during a strenuous workout or sudden sprint to catch a local train. Next, we come to the hemoglobin molecule. It is the oxygen distributor. Hemoglobin is the one which resides within red blood cells. It is a tetramer and it is composed of 4 globin subunits, 2 alpha and 2 beta, each carrying its own heme group. This structure, like a 4-compartment tiffin box, allows for cooperative binding, which is a unique property that makes hemoglobin a highly efficient oxygen transporter. The binding of the first oxygen molecule induces a conformational change in the entire hemoglobin molecule, making it easier for subsequent oxygen molecules to bind. This is like opening one compartment of your tiffin box, making it easier to open the others. This cooperativity results in a sigmoidal oxygen binding curve, allowing hemoglobin to load up on oxygen in the lungs, where oxygen partial pressure is high, and release it efficiently in the tissues, where oxygen partial pressure is low. Then we come to the concept of allosteric regulation, which is the fine-tuning the oxygen express. Okay, so this allosteric regulation fine-tunes the oxygen express. So hemoglobin's oxygen affinity isn't static. It's dynamically regulated by factors like pH, CO2, and 2,3-bisphosphoglycerate. These allosteric regulators, they act like signalmen controlling the oxygen express. They fine-tune oxygen delivery to match tissue demands. A decrease in pH is the Bohr effect, increased CO2 levels, and 2,3-BPG all decrease hemoglobin's affinity for oxygen, promoting oxygen unloading in metabolically active tissues. Think of it like this: when you exercise, your muscles produce more CO2, carbon dioxide, and lactic acid, which lowers the pH. This signals the hemoglobin to release more oxygen, fueling the working muscles. In high altitude areas, 2,3-BPG levels increase, facilitating oxygen delivery to tissues despite the lower oxygen availability in the environment. Then we come to a concept of comparative biochemistry and oxygen management across species. Interestingly, different species have evolved unique hemoglobin adaptations to suit their environments. High altitude animals like the yak have hemoglobin with a higher affinity for oxygen, allowing them to thrive in oxygen thin air. While deep diving mammals like whales have higher myoglobin concentrations in their muscles, enabling them to store larger amounts of oxygen for prolonged dives. Now let's just see a couple of clinical connections, okay? So the first clinical connection associated with hemoglobin is anemia. It's a common condition, especially in India, particularly among women and children, which often results from iron deficiency, leading to reduced hemoglobin production. This results in decreased oxygen carrying capacity, leading to fatigue, weakness, and pallor. Then we come to something called sickle cell anemia. This is an inherited disorder. It is prevalent in certain parts of India and is caused by a mutation in the beta globin gene, leading to the formation of abnormal hemoglobin, which is also called HbS. This HbS polymerizes under low oxygen conditions, causing red blood cells to become rigid and sickle shaped. These sickle cells can block blood vessels, leading to painful crisis and organ damage. And we come to thalassemia, which is another prevalent inherited blood disorder in India. So, thalassemia involves reduced or absent synthesis of one or more globin chains. This imbalance in globin chain production leads to ineffective erythropoiesis and anemia. Different types of thalassemia exists with varying severity depending on the specific globin chain affected. We won't go into the depth of that. Then we come to carbon monoxide poisoning. So carbon monoxide it binds to hemoglobin with much higher affinity than oxygen, forming carboxyhemoglobin. This effectively blocks oxygen binding and transport, leading to tissue hypoxia. So, poisoning can occur from exposure to faulty heaters or exhaust fumes, and it's crucial to recognize the symptoms, which include headache, dizziness, and nausea. So, remember the four subunits of hemoglobin as alpha, beta, gamma, delta, like the different variants of COVID 19. Although HbA is alpha 2, beta 2. Now, think of 2,3-BPG as Bohr booster. It enhances the Bohr effect by decreasing hemoglobin's affinity for oxygen. Finally, just to summarize all of it, hemoglobin and myoglobin are essential for oxygen transport and storage. Heme, containing iron, is the oxygen binding site. Myoglobin is a monomer and it stores oxygen in muscle. Hemoglobin is a tetramer and it exhibits cooperative binding and transports oxygen in blood. Oxygen affinity of hemoglobin is regulated by pH, CO2 and 2,3-BPG. Clinical conditions like anemia, sickle cell anemia and the thalassemia highlight the importance of these proteins. So now you know about hemoglobin and myoglobin. So I hope to see you in the next one. Thank you for listening.